Assalamu alaikum. So now that we learned the structure of hemoglobin and how it's regulated by certain allosteric effectors like oxygen itself, um, protons, CO2, bisphosphoglycerate, chloride ions, and so on, and carbon monoxide. And we talked about certain amino acids within the protein, amino acid residues that regulate the function of hemoglobin. Let's talk about hemoglobinopathies. So, what are hemoglobinopathies? So, hemoglobinopathies are basically disorders of human hemoglobin. Basically, we have deficiency in the oxygen transport system. Now, these are really serious diseases as a group because they are the most common. And we have 300,000 infants born each year with a hemoglobinopathy. And they account for about 3.4% of deaths in children below 5 years old. So we're talking about something that is quite serious. Now, notice here in the map, that's the distribution of hemoglobinopathies around the world. And notice that these are concentrated in the old world. The Middle East has a significant share of hemoglobinopathies. So this is something serious, and that would include Jordan as well. So we can classify hemoglobinopathies into different types. We have what is known as qualitative abnormalities. So basically we have mutations that result in different structure of hemoglobin that makes it defective in transporting oxygen. So each amino acid has a function in a hemoglobin molecule. So and there is a good chance that one of them would be mutated, and as a result that would again cause the production of a defective hemoglobin. So we have over 700 variants of hemoglobin molecules because each one of them can be mutated again. So these are known as qualitative abnormalities or qualitative hemoglobinopathies. We also have quantitative abnormalities. Basically, the molecule is fine in structure except that there are less quantities of hemoglobin molecule, whether alpha chain or beta chain, in cells. And these are collectively known as thalassemias. Now, Jordan has a high prevalence of thalassemias. Now, we have a third type of a hemoglobinopathy, and it's known as hereditary persistence of fetal hemoglobin, where basically there is no switching from gamma to beta globin. So we'll talk about this at the end. So let's start with qualitative abnormalities. So basically we're talking about single point mutations, mainly single point mutations. Now these mutations can take place anywhere in the hemoglobin molecule. So they can occur on the surface or they can be internal. Now if they occur on the surface, usually they are asymptomatic. An example is HBE, and we'll talk about it in a few minutes. Now, an exception is HBS, or sickle cell anemia. This is a single point mutation that really cause dramatic effect on the function of hemoglobin molecule. Now, mutations can also occur internally. Now, remember, hemoglobin is a globular protein, so you have hydrophilic amino acids on the outside and you have hydrophobic amino acids inside. Now, if this environment is changed, then that would cause the production of an unstable hemoglobin molecule. An example is hemoglobin Hammersmith. Now you can also have mutations stabilizing the methemoglobin, which is basically a hemoglobin with iron in the ferric state rather than the ferrous state. usually that results in cyanosis since this hemoglobin cannot bind to oxygen. Now, you also have common mutations in the alpha 1 beta 2 contacts. Now, that would cause an alteration in the equilibrium of the T hemoglobin and R hemoglobin. And that would cause, again, altered oxygen affinity, which is a condition known as polycythemia. So let's start talking about sickle cell hemoglobin. So what happens here in this hemoglobin molecule, and we're specifically talking about the beta globin chain, is that there is a mutation in the 6th position in amino acid number 6 in the primary structure of the protein. The mutation is basically a glutamine to valine. Okay, so glutamine is what? It is a negatively charged amino acid. It changes to a non-polar amino acid. So that basically results in a change in the structure of the whole polypeptide. So that change leads to protein aggregation, especially when the protein is in the deoxygenated state. Okay, so instead of having this nice structure, rather you have a change in the structure that leads to clustering or aggregation of the protein. So instead of having separate individual proteins, you have this aggregate right here. Okay, now this aggregate leads to a change in the shape of the cell, the whole cell. So instead of having this nice concave structure of the red blood cell, rather you have a sickled structure or sickled morphology or shape. Okay, now that leads, by the way, to a reduction in the lifespan of the cell. So instead of having a lifespan of 120 days, it becomes less than 20 days. That leads to disruption or death of the red blood cell, premature death of the red blood cell, and leads to what is known as hemolytic anemia. Okay, now the protein itself is designated as alpha 2 beta S2. Okay, S stands for sickled, or the whole molecule is designated as HbS. So what happens here is that with repeated cycles of oxygenation, deoxygenation, that would cause clumping and clustering of the hemoglobin molecules. Okay, now it would look something like this. So you have an array of molecules clustered to each other. Now that would cause clumping into capillaries, clogging capillaries, so that blood flow is restricted, causing hypoxia, low level of oxygen in tissues. Now eventually that would lead to organ damage, particularly in kidneys, heart, and lungs. So the question now is, why is it that hemoglobin molecules, or the sickled hemoglobin, cluster together? See, what happens here is that right here you have a hemoglobin molecule that is oxygenated. It looks like this. Okay. Now, if it is deoxygenated, it would cause the formation of a hydrophobic pocket within the molecule itself, within the beta 1 chain. Okay, so in a sickled hemoglobin, you have the formation of a valine protrusion or a hydrophobic protrusion that includes a valine molecule. Now this is the sickled hemoglobin right here. So when it becomes deoxygenated, this protrusion still exists, but you have the hydrophobic pocket in here. So what happens is that this protrusion would fit into this, of one molecule would fit into the pocket of another molecule. So here you have the hydrophobic pocket right here. That's the protrusion, and the protrusion would fit into the hydrophobic pocket. So right here you have the valine protrusion, and it gets inserted into the hydrophobic pocket. Okay, so that would cause clumping of the hemoglobin molecule. So remember that I said that sickling increases with repeated oxygenation deoxygenation cycles. So there are certain variables that increase sickling. One of them is decreased oxygen pressure, which means that you have a high probability of having a molecule that looks like this. Okay, same thing for the other factors. So you have increased the pressure of CO2, decreased pH. That would also increase the formation of the T structure of hemoglobin. Increased 2,3-bisphosphoglycerate, same thing, stabilizing the T structure, and dehydration as well. Now, there is something known as sickle cell trait, which is basically individuals that are heterozygous, meaning that they have both HbA and HbS. This is a case where, again, one allele is normal, it's HbA, producing normal beta chain, and the other one is mutated so that you have the formation of HbS. So you have a combination of HbA and HbS. So this is a normal red blood cell, for example. This is a cell that contains high quantity of HbS. Okay. Now, so these individuals actually do have an advantage. Okay. Now, the advantage is that individuals who are heterozygous, who have a sickle cell trait, is that they become resistant to infection by malaria. The reason is that you have one reason is that you have lower half-life of red blood cells, so that they are less than they're about 40 days or even less than that. Now, the half-life or the reproductive duration of malaria is about 60 days. So, and malaria infects red blood cells. So that would decrease. So having lower half-life of red blood cells would would prevent malaria from completing its growth inside cells. So these individuals are resistant to malarial infection. And if you look at the distribution of sickle cell allele in Africa, you can see that it actually matches the distribution of malaria in these countries. So it fits. So there's an advantage, a genetic advantage for these individuals. So another quantitative hemoglobinopathy is known as HBC, where basically you have a single point mutation that changes the glutamate at the 6th position, which is exactly the same as sickle cell anemia or HBS, except that it does not change to valine, it changes to lysine. And this beta globin is known as beta C. Now, this hemoglobin is less soluble than HbA, so what happens is that it crystallizes in red blood cells, reducing the ability of red blood cells to change shape as they squeeze in capillaries. Okay. So, for some reason, HbC leads to water loss from cells, leading to higher hemoglobin concentration again in these cells. Now, remember how I said that changing the amino acids on the surface of the hemoglobin molecule it has insignificant effect on the function of the protein. Well, that's exactly the case here. So contrary to HBS or sickle cell anemia, having HBC causes only a minor hemolytic disorder. So if you look at the previous slide, actually you can see the abnormality in the morphology of red blood cells in individuals having HBC. Now there's another condition known as HBSC. Now these individuals have two alleles with different mutations. One allele is actually a beta S and the other allele is a beta C. Now So they produce defective hemoglobin molecule. Now, just like normal individuals or individuals with sickle cell trait, these individuals have mild clinical abnormalities, hardly any clinical consequences, but it is clinically variable. Now, there's another condition known as hemoglobin E. Now, this condition is common in Southeast Asia. So if someone has a servant, a housemaid coming from Southeast Asia, there is a good chance that this person would have HBE. Now, these individuals have here, they do have a single point mutation in the globin molecule in beta globin, except that now the change is actually on position 26. That would change glutamic acid into lysine. Now, there are two consequences of having this mutation. The first one is genetic. So basically what happens is that there is that would create an alternative RNA splice site, leading to the production of a defective protein. Now, this protein also, once formed, it's not as stable, of course, as the normal protein, and it would tend to be degraded. So individuals, not only that they have deficient ability of hemoglobin to carry oxygen, they also have lower amount of the beta globin protein. So this condition has both quantitative and qualitative characteristics. Another condition is known as HB-Hammersmith. Now, here in this condition, we have a point mutation that changes a phenylalanine into a serine. Now, why is that important? Because this phenylalanine exists in the heme pocket. So it interacts via hydrophobic interactions with the heme molecule, stabilizing the positioning and the interaction of heme into, the positioning of heme into the hydrophobic pocket of hemoglobin molecule. Now, if this phenylalanine changes to serine, that would cause destabilization of the interaction between heme and the hemoglobin molecule. That would cause unstable hemoglobin molecule, denaturation of the globin protein, inability of the globin molecule or hemoglobin molecule to bind oxygen, and that would cause ultimately in cyanosis. Now, there are mutations that can alter the stabilization of the T and R state. Now, if you go back to the previous lecture, we talked about how certain amino acids can form electrostatic interactions, hydrogen bonds, stabilizing either the T state or the R state of the protein. So we'll talk about three variants here in this slide. We'll talk about a variant known as hemoglobin cow-town. That variant does not have histidine 146. Now remember, this histidine is the very last histidine in the beta globin polypeptide. It's the one responsible for the Bohr effect. It forms an electrostatic interaction with both lysine on the opposite chain with the carboxy terminus, and the imidazole ring forms an electrostatic interaction with aspartate on the within the same polypeptide. Now this right here, this interaction results in stabilization of the T state. Now if you don't have this histidine, then these interactions cannot take place, electrostatic interactions, and you have the protein in the R state. So the protein has very high affinity for oxygen, and as a result, when it gets to tissues, peripheral tissues, it does not release oxygen easily. Now there are other variants as well. You have a variant of hemoglobin known as hemoglobin kansas. Now, in this case, this asparagine is altered to threonine. Now, this asparagine stabilizes the R state by forming this hydrogen bond between asparagine in the beta chain and aspartate. So you don't have this formation of hydrogen bond, and as a result, the protein would exist in the T state because the R state is not stabilized. Now, you have another variant known as hemoglobin Yakima. In this case, this aspartate is altered to histidine, and as a result, the histidine cannot form electrostatic interaction with histidine 146, the last histidine in the beta globin chain, and as a result, the T state is not stabilized, and you have the protein in the R state more frequently. So in this case, again, the hemoglobin binds oxygen, but it does not release it in peripheral tissues. So these are some examples of qualitative hemoglobinopathies. Now, remember, there are 700 variants, so we can talk about every single amino acid, and there are different names for different hemoglobin variants. Let's talk about another condition or consequence of qualitative abnormalities, which is known as altered oxygen transport. So we talked about methemoglobin briefly, which is basically a hemoglobin molecule that is bound to iron in the ferric state rather than the ferrous state. Now remember that this hemoglobin molecule, which is known as methemoglobin, is not able to bind oxygen. Now, do normal people have methemoglobin? Yes, they do, but the quantity is very low. The reason is that, well, there are two reasons. The first reason is that the hydrophobic pocket surrounding the heme molecule prevents the oxidation of iron from the ferrous to the ferric state. Okay, so what happens is that you have electron rearrangement between the different amino acid residues, preventing this oxidation from taking place. But the other reason is that individuals have what is known as a methemoglobin reductase. See, what happens is that if you have the formation of a methemoglobin, immediately this reductase, the methemoglobin reductase, what it does is that it immediately reduces hemoglobin, methemoglobin, to a hemoglobin molecule with iron in the ferrous state. This requires cytochrome containing cytochrome molecule within the enzyme itself, and that requires NADH as well. Okay, so you have oxidation of NADH and transfer of electrons to eventually to iron. Okay, this NADH, this reaction takes place, by the way, in red blood cells, of course. Okay, and this NADH, the main source of NADH is glycolysis, and we will talk about this in the next lecture. So there's a condition known as methemoglobinemia, and in people with this condition would have their blood looking like chocolate brown, and they have chocolate lips, okay, because of cyanosis. Now, how can these individuals be treated? Now, there are certain variants, certain mutations in hemoglobin molecule that can result in the formation of the methemoglobin. Okay, so these mutations either occur in the alpha or the beta, and there are so many of them. Okay, so what these mutations do is that they can affect the status, the oxidation status of the iron. Okay, we'll talk about the two of these variants, and both of them occur in the alpha chain. So the first variant is known as hemoglobin Boston. Now, in this case, you have a mutation whereby the distal histidine is mutated into a tyrosine. The distal histidine, if you remember, it functions as a gate. It allows for oxygen as well as carbon monoxide to enter into the hydrophobic pocket and bind to heme. Okay, but these mutations prevent other molecules like water from getting in. So if you have a mutation in the distal histidine, in this case, well, tyrosine would exist in front of the heme, and what it does is that it stabilizes the ferric form of heme or iron, and that's methemoglobin. Now there's another variant known as HBM-EWATI, whereby the proximal histidine the proximal histidine is the one that forms a covalent bond with the heme, with the iron. Now the proximal histidine is mutated into a tyrosine. Again, tyrosine can bind to iron, except that it would stabilize the ferric form rather than the ferrous form. Okay, so in this case, in both of these variants, the hemoglobin, or heme, I should say, cannot bind to oxygen. Now, you can also have deficiency of in the reductase enzyme itself. Now, by the way, these mutations would resist the reductase activity. Okay, so the enzyme itself cannot reduce iron from the ferric to the ferrous state. Or you can have deficiency in the reductase enzyme itself. And there are certain drugs, as well as if drinking water contains nitrates, that would also result in it favors oxidation of iron into the ferric state and the inability of heme or iron to bind to oxygen. So, as I said, normally what happens is that NADH from glycolysis is the main source for electrons that would be donated ultimately to iron plus 3, converting into Fe plus 2. Okay, so an alternative pathway can be used utilizing methylene blue. Now, there are two major metabolic pathways in red blood cells. One of them is glycolysis, and the other one is pentose phosphate pathway. So what happens with the pentose phosphate pathway is that you have the production of NADPH. Now, NADPH is the source of electrons that would be used to reduce methylene blue to another product known as leucomethylene blue. Now, leucomethylene blue can be used as a source of electrons for methemoglobin, resulting in the production of the normal globin molecule. So methylene blue can be used in treating people with methemoglobinemia. So let's discuss now quantitative hemoglobinopathies, which again, they're known as thalassemias. So thalassemias are the most common human single gene disorder. They are quite prevalent in the old world, including the Middle East. And basically these are caused as a result of disruption in the alpha to beta ratio. So in red blood cells, what you have is equal amount of alpha chains and beta chains. So in thalassemias, you would have either more alpha than beta or more beta than alpha. Let's start with the alpha thalassemia. So alpha thalassemia means that there's underproduction of the alpha globin chains. So basically you have more beta than alpha. Now remember that the alpha chain is produced at the end of the embryonic stage. Okay, so it starts early on and it continues throughout life. On the other hand, the beta chain starts to be produced slowly in the early fetal stage, but there's a big jump right before birth, and it continues throughout life again having again equal quantities of alpha and beta. Now, if there is low production of the alpha chain, this means that there is a high probability that you will have a hemoglobin molecule that is composed of 4 chains of beta. Such molecule is known as HBH. So an HBH is a hemoglobin molecule that is a homotetramer of beta. Now, HBH molecule has reduced oxygen carrying capacity. The main reason for why a person would have alpha thalassemia is deletion mutations rather than single point mutations. Now remember that we have 4 alpha genes. We have 2 on each chromosome. So we have a total of 4 alpha genes. So as a result, normal individuals would have all 4 of them being active. There is a probability that one of them would be mutated. So you would have 3 genes that are active, or you can have 2 that are deleted, or 3, or all 4. That would result in variable severity. So for example, you can have people with only 1 gene deleted. These are known as silent carriers. Okay, so they do not complain from anything. Now, if 2 are mutated, this is known as a alpha thalassemia minor. If you have 3, then this is known as HBH disease. That's because individuals would have a high quantity of HBH hemoglobin, and it becomes very severe if all 4 alpha genes are deleted. Now, if 4 alpha chains are deleted, then you get a condition known as hydrops fetalis. Now, in this case, you will have a fetal hemoglobin that is a tetramer of the gamma chains. Now, remember that the alpha chain is produced early on in the start of the fetal stage, and it continues throughout life. So you have a hemoglobin that is predominantly a homotetramer of gamma. Now this HB BART has no oxygen capacity, okay, meaning that meaning that it is a bad oxygen carrier. Now it's not because it doesn't bind to oxygen, rather it does bind to oxygen, but it has high affinity for oxygen, that to the extent that it cannot release it in tissues. Okay. Now what happens is that this causes stillbirth or death shortly after birth because oxygen cannot reach tissues. So you have starvation, oxygen starvation in fetal tissues. Now, if 3 of the alpha chains are mutated, or the genes are mutated, then that would cause a formation of a beta-4 tetramer, homotetramer. Now, this is known as a condition known as hemoglobin H disease. Okay. Now, there is some alpha, but not a whole lot, and that's why the hemoglobin would predominantly be a beta-4 homotetramer. Okay, now, or what is known as HBH. This hemoglobin molecule is known as HBH, and that's why the disease as a whole is known as the hemoglobin H disease. Now, the disease is not fatal. Okay, the person survives, but this person does have a poor ability to supply peripheral tissues with oxygen, because HBH, just like the HB BART, it has high affinity for oxygen because it does not have a Bohr effect. So it has very high affinity, and oxygen is not released in peripheral tissues. Now you have minor alpha thalassemia, where basically 2 genes are active. Now these individuals are asymptomatic. and you have a silent carrier where one gene is deleted. These people are totally asymptomatic. So these are again the different possibilities. Normal individuals have all four alleles being active. Here you have in silent carriers, you have three of them being active. And thalassemia trait is when two of them are active. So you can have deletion of the alpha alleles on one chromosome or one from each chromosome. So this is known as thalassemia minor or thalassemia trait. Now the HbH disease, people have mild to moderate symptoms, and in hydroxyphthalate you have, which is known as thalassemia major, you have all four of them being deleted. So how about beta thalassemias? Basically, in this condition, results from defective beta globin genes. Now remember, normal individuals have two alleles, one on each chromosome. So mutations basically result from are basically point mutations rather than deletion mutations. So you can have different types of deletions within the gene itself, the coding region that codes for proteins, or it can be in the translation initiation codon, the splicing condition, splicing positions, or polyadenylation termination signal. Okay. Now that would result in the formation of a alpha globin homotetramer, globin. Now this protein is extremely insoluble, and that would result in destruction of red blood cells. Now, there are different types of beta thalassemias depending on the severity of the mutation. So if there is complete lack of HbA, that would result in what is known as beta not thalassemia or beta thalassemia major. That is, there's no production of the beta globin at all. Now, these individuals do suffer from anemia beginning from the first year. These individuals would need transfusions, blood transfusions. The problem with blood transfusions is that there would be accumulation of iron in organs, and we'll talk about metabolism of iron at the end of this series of biochemistry lectures, and that would cause tissue damage and organ damage, particularly liver and pancreas. Eventually that would lead to death in the early 20s. You can also have individuals with beta thalassemia minor, meaning that there is some production of the beta globin. These individuals are heterozygous for beta thalassemia. Okay. Now, they have one normal beta globin gene or two that produce low amount of beta globin. Now, usually these individuals are asymptomatic. So there are different types of beta thalassemias. So normal individuals have normal alleles of beta globin on both chromosomes. Now you can have one of them normal, but the other one can be mutated or defective. This is known as beta thalassemia trait or thalassemia minor. These individuals are asymptomatic. Now you can have different combinations as well. So you can have both alleles producing little amount of beta globin. You can have one of them being mutated altogether, not producing any beta globin at all, and the other one is defective, producing some. You can have individuals with HBE, okay, but the other allele is mutated, producing little amount of normal beta globin, or you can have these individuals as well. So this is known as beta thalassemia intermedia. They have variable severity, they have mild to moderate anemia, and so on. But they have iron overload. Or you can have both of these alleles mutated, producing very little or producing no beta globin at all. and this is known as beta thalassemia major. These individuals have severe anemia. They require transfusion frequently, resulting in what is known as iron overload. Now, the final condition is known as hereditary persistence of fetal hemoglobin. Now, in this case, these individuals have an abnormality in not transitioning from gamma to beta during development. Okay? That would result in individuals producing HbF throughout their life. Now, these individuals do not know that they have an abnormality whatsoever because HbF is still functional and it's good. Now, yes, HbF can bind to oxygen with higher affinity than HbA, but that doesn't really give an advantage to these individuals. Now, the beauty of this, remember when we talked about genetics of the globin synthesis? Well, and I told you, I gave you a hint that this can be used as a treatment for beta thalassemia. Well, individuals who do not produce any beta globin, why don't we let them produce gamma globin? Okay, so there is an idea of switching on the fetal globin gene in case the beta globin gene is not functional. So there's talk about that. This is taken from a good journal known as Nature Genetics in 2018. Okay, so let's talk about diagnosing hemoglobinopathies. This can be done using electrophoresis. We talked about this before in our biochemistry lectures, and we said that this is a technique whereby molecules such as proteins can be separated according to size, they can be separated according to charge, and so on. So what happens here is that the same thing can be done with hemoglobin molecules, especially those that have single point mutations whereby you have a change of one amino acid to the other. This is particularly true if the amino acid that is changed or that is added would change the net charge of the molecule. So changing glutamate, a negatively charged amino acid, to a valine or to a lysine would change the overall charge of the whole molecule. And as a result, hemoglobin molecules would travel differently. Okay, so this electrophoresis technique depends not on the size of the hemoglobin molecule, rather it depends on the charge of the molecule. So you can read this on your own. Let's look at an example of an electrophoresis result. So basically what you have in here is different samples. In number one, lane number one, we have a standard, meaning that, same thing with number 5, meaning that it's a sample that contains all forms of hemoglobin. So you have HbA, HbF, HbS, and HbC. Now you can see the migration pattern is different among the different hemoglobin molecules. So you have the HbC, for example, traveling close to the cathode, negative charge, and HbS is migrating a bit slower than HbC, and so on. So why is it the case? Why is it that you have this different migration pattern? Well, remember with C, what you have is a mutation of glutamate to lysine. So you have a loss of one negative charge and an addition of one positive charge, and that's why it travels closer to the cathode. Now here you have, with HbS, you have loss of a negative charge, glutamate, to valine, okay, which is no charge. Same thing, the molecule would travel away from the anode and closer to the cathode. Now HbF as a whole molecule has it's more positively charged than HbA, the adult hemoglobin. So these are that's hemoglobin or samples taken from different individuals. You can see that this individual, for example, has only HbA. So this is an adult individual. Look at this individual right here. Sample number 3 is taken from a person having both HbA and HbF with more HbF than HbA. So this is an individual who is a neonate, most probably. Sample 4, HbS only. So this is someone with sickle cell anemia. Look at number 6. You have both HbA and HbS. So this is someone with sickle cell trait. This is number 7, an individual with HbSC. And individual 8 is someone with with sickle cell sickle cell trait as well. So that's how it looks like.